Okay, awesome biology kids. We are ready for the lab of the microscope lab for the cell cycle and onion root tip. So here's the directions. You should have already printed these out. It says a microscopic view of the cell cycle and allium root tip. So the supplies we need for this lab are kind of minimal. In fact, here's the slide that we're going to be using. You can see if you look closely, the black little lines or bluish looking lines that you see right there, those are little slivers of the allium, which is the type of plant, and it's off the root, the tip of the root, because we know that the roots are growing, which means there's a high likelihood that we're going to see some cells that are actively going through the cell cycle. So it gives us more than just the typical interphase. We should be able to see some good specimens that are in prophase, metaphase, anaphase, telophase, and cytokinesis. So we need that slide. We also need a pencil so that you can draw in pencil. And you need this answer document. So make sure you've printed out the answer document. There is a analysis section on the back. Okay, so you need to have this. And we're ready to go. Okay, so the first thing that we're gonna do is we're gonna make sure that our microscope is on low power. So you guys can see right here, it's the 10X. It's got a yellow band at the top. So we're gonna make sure that's clicked in. You can see right here how it does a nice click like that. Make sure it's clicked in. We want to use the coarse adjustment knob, this big knob, and make sure the stage is all the way down, as low as it can go. All right, now with our slide, we're gonna carefully place the slide underneath the stage clips and make sure we secure it. We're not using one of the fancy microscopes this time. We're using the ones with the regular stage clips. So we're going to center what we see with the root tips over the field of view. And now we're ready to turn on our lights, get a little illumination going, and we're gonna focus it. So to focus, we're gonna use the coarse adjustment knob, which I always call this the fat knob or the big knob, and we're gonna look through the eyepiece. Remember, the eyepiece has a 10X objective. It multiplies 10 times. So if we're looking at this on the low power, the 10X, remember we multiply, the eyepiece is 10, the objective is 10, 10 times 10 is 100. So we're really gonna view these cells on 100 times. So I'm going to use the coarse adjustment knob. While I look through here, I'm going to raise the stage. You can see it coming up like that. I'm going to raise it until I find what I'm looking for. Okay, so I found a pretty good area. I'm going to try to focus it so you can see. You're not going to make your drawings by looking at this. I just want you to be able to see what I'm looking at through the microscope. Hang on, give me a second. Un momento. Hang on, hope you're not getting dizzy. I'll let you know when I got it. Hang on. All right, there we go. Now I know it's not very focused, but do you see how that goes to a point? It's like an arrow at the bottom. Don't pay attention to the black line. The black line is my pointer. But do you see how, oh, that's a pretty good, it just focused. There we go. Do you see how all those little boxes, little circles that you see on there, those are each individual plant cells. And the tip is going to, because that's the root that's growing down in the ground. So that's on low power. So we want to see this on high power. So give me a second. And I will come over here, and we're going to turn the revolving nose piece, which is this. We're not going to touch any of the knobs because it is focused, and we're going to turn this so that the 40 clicks into place. And I'm going to show you on this side. There's the 40. So this is 400 times, and we're going to look through here, and if it's fuzzy, this is when we're going to use the fine adjustment knob, the small knob, just to fine tune the image. Okay, so I have it focused on the 40, which is the 400 times. So I'm going to try to get a view for you to see in here. Ooh, look how nice those are. Hang on, let me focus it a little bit. Okay, check that out. So you're seeing a lot of different cells in different phases of the cell cycle. So it's hard for you because you're kind of watching this to pick some out. So I just want to show you so you can get your bearings for what you're looking at. If you look in the row, oh, there we go, it just came into view. If you look down the row where the black line is, if you count down about 1, 2, 3, 4, 5, 6 cells from that, you will see a beautiful metaphase. And you can see because it has the DNA is all kind of stringy lined up in the middle of a cell. That's a pretty good specimen of a metaphase. If you look right below the pointer, like the one right below, not the one it's on, but the two, there's like 2 and 3 and 4 lined up. Those are all interphase. Most of these are in interphase. Okay, but I know it's hard for y'all, so what I have for you, I just wanted to show you as I was focusing it. I have some pictures that I'm going to lay on the table that you can use to make your drawings. All right, so here are the cards that I want you to use. I'm gonna focus in on them. These are really good, by the way. Look, I'll get it close up like this. So you can pick out, make sure you use your notes so that you know which phases you're looking at. So you're gonna try to find one that's interphase. Keep in mind, interphase, you should be able to see the nucleolus, okay, nucleolus. Prophase, you should see DNA is kind of like coiling and condensing and the nuclear envelope is disappearing. With metaphase, you should see the DNA lined up in the middle. Anaphase, they should be pulling apart. And then telophase and cytokinesis, you should see the formation of a cell plate, because these are plant cells, these are not animal cells. So there's another field of view for you. Lots of choices for you guys on these to use. Okay, that one's got a lot of interphase in that one. Okay, look at this one. I see a good prophase in this field of view. See if you can find it. I see a really good metaphase. I see probably the best telophase cytokinesis near the top. That one's amazing. Okay, look over here. So you get to choose from all of these to make your drawings. So what you want to do now is find, you know, push pause, I guess, on the video, find some good representative specimens, and you're going to make a drawing. Start here with interphase, pro, meta, ana, and then this one is telophase and cytokinesis. So when you make your drawing, I do want you to draw what you see. Do not make anything up from the notes. You're not gonna see extra stuff, so you're really just drawing what you see. This is one cell, like right here. And then make sure you put your drawing off to the left, like we did on the last lab, so you can use a ruler and label off to the right. Now, what do you label? Look right here in the directions. You're going to label these things if they're present. Okay, so like on the first one, DNA. Well, I can tell you DNA is present in every single phase, so make sure you have that labeled, but I want you to specify, is it chromatin, is it sister chromatids, or is it daughter chromosomes? So you're gonna choose one of those to go with the DNA label. Okay, nucleolus, label it only if it's there. Like hint, hint, in metaphase, there is no nuclear envelope or nucleus left, so you should not be labeling nucleolus in metaphase. Okay, nuclear envelope, same thing. You're not going to see it in metaphase. Cell wall, well, every cell, every picture should have a cell wall because they're plant cells. Cell plate, that should be visible in cytokinesis, right? So you're not going to see that in interphase or prophase, etc. So you're going to label those things. I do want you to shade them the color that they appear with colored pencils. So obviously you can tell it's more of a like bluish purple. I see a little bit of pink, a little bit. So you're going to shade the color that you see. Once you're done, and then after that, you turn it over and answer the analysis section. Okay, that's it.